All right, anyone out there, stream should be live now, and you can probably see my desktop and me hiding in the corner. We'll start in just about a minute or two. All right, guys, we're going to go ahead and get started. Hello to everyone out there in AP Biology land. My name is Helen Snodgrass. I teach AP Biology in Houston, Texas. And I'm super excited to be with you guys today on the last day before the AP exam. I'm going to try to give you some last-minute strategies and advice to help you do your best tomorrow. Got to admit that I am old and not very good with technology, so I apologize if there are any issues, but we've got some moderators out there helping me out, so we will get anything fixed if there are any problems. All right, so let me give you guys just a few logistics so you kind of know what the session is going to be like, and then we will get started. First thing, as I said, technology, there could be issues. I hope there are not, but we will fix them if there are. And just so you know, this session, excuse me, as well as all of the other review sessions have been posted online. This one will be available as soon as it is finished, so you're welcome to go back and watch anything that you would like to. A lot of you submitted questions ahead of time that helped us plan this review, so I've already seen a lot of those questions. Thank you for that. But you can also submit questions in the chat box during the session. I can't currently see them, but we have some moderators out there who are going to answer some of those questions and also send some common ones my way so that if there's anything we want to add or clarify, I can do that. You guys also probably have possibly even better ideas than I do for strategies and tips for the exam. So if there's anything you think of that has helped you really well in AP Biology this year, please share those out in the chat box and we'll get some of them on here. All right, so for exam strategies, that's what we're going to be spending the majority of the time on. These are all just suggestions. You do not have to do all of these things. There's probably some that you've tried and things that work for you and things that don't. So definitely take them as suggestions. If we have a little bit of time at the end, we might review some content if some questions come up. But again, all of the review sessions that have already happened are all available on YouTube. Those cover all the big ideas as well as quantitative skills and labs. So definitely check those out if you have some other questions. It would help you to have some paper and a pen or pencil out. We are going to look at a few practice questions as we think about strategies. And we already said that. Last thing is just a quick thank you to all of the teachers out there who helped organize these reviews, have led the reviews, or moderating them, as well as to all the people who've shared a lot of the strategies that I'm going to share with you today. I definitely cannot take credit for having thought of all of them or most of them, so shout out to all of the amazing AP Bio teachers out there. All right, so for our agenda, we got a number of questions about mindset and how to deal with stress about the exam, so we're going to start out talking about that. share a few tips for studying and overall thinking about the exam, although there's not a lot of study time left. We'll specifically look at the multiple choice and free response and look at some example questions. And then again, if there's a little bit of time, we might do some review of just general content questions that come up at the very end. All right, so my biggest piece of advice, which I know is much easier to say as a teacher who's not going to be taking the exam tomorrow, but I have taken many big tests in my life, including AP Biology, is that this is just one test. I don't say that to say that the exam is not important, that you shouldn't care about it. If you're out there watching this on a Sunday, you obviously care about it, and you should. You should do justice to all of the hard work you've done this year by trying your best. But there's absolutely no way that one single exam could possibly capture all of the incredible amount of information and skills that you've learned this year. So no matter how the exam goes, you should be really proud of yourself for that. And a shout out to your teachers. You should thank your teachers for all that they've helped you with this year, no matter how you feel after you take this test. All right, a few more specific suggestions. So today is your very last day to study if you're taking the exam tomorrow, which most of you out there will be. And my best piece of advice is to not try to spend the entire day, definitely not the entire night, studying. Most of the things that you need to know for the exam, you already know, and a lot of last-minute cramming isn't necessarily going to make a big difference. So spend some time relaxing. And definitely get plenty of sleep Sunday night. You probably heard this advice a million times, but sleep does two really important things for you as far as getting ready for taking a big exam like this. One is that it's going to help you consolidate any information you learned during the day. So if you do some studying today, you want that information organized and accessible for you tomorrow. Second thing it's going to do is it's just going to help you think clearly during the exam tomorrow. And if you've done any practice tests, you know that a lot of the AP Biology questions are very complex, require you to do some deep thinking. So you want to make sure you're prepared to do that. You also want to basically follow your normal routine tomorrow morning. By that I mean if you normally drink coffee, go ahead and have some coffee. But if you are not a regular caffeine drinker, don't think that having a whole lot of caffeine is really going to help your performance. It's actually better just to stick with whatever you normally do throughout the year, whatever your normal routine is before school. One last piece of advice that's actually been studied in a number of tests. I included some links for you there if you're interested in looking more at it, is to take 5 or 10 minutes either tonight or before the test Monday morning if you have time to write. The two things you should write about: one, if you're feeling anxious about the test at all, write about what's making you feel anxious or stressed out. Might seem counterintuitive, but actually writing about what's making you feel stressed can actually make you feel less stressful about it. The other thing to write about is what are your values? What is most important to you as a person? That's just going to help ground you in the fact that this is just one test. There's a lot more to what is important to you as a person. Hopefully help you let go a little bit of some of that anxiety. Last tip here is just keep in mind as you go through the test that there's going to be questions that feel extremely challenging and that you feel like you do not know the answer to at all. That's okay. You do not need to get anywhere near all of the questions right, either on the multiple choice or the free response, to do very well. So don't be concerned if you feel like there's some things that are really hard. You should expect there's to be some things that are really challenging. Okay. All right, now we're going to take a look at a few overall study and exam tips. So if you are going to do some last studying today, which again, I don't recommend studying from now until midnight tonight, but if you're going to study a little bit, do focus on the big concepts. We got some questions about whether you should spend a lot of time on the details of little processes. Most questions are going to be grounded in your knowledge of bigger concepts, and given the limited time you have right now, that's probably the best place to focus. And in terms of trying to figure out which big concepts to focus on and what is included in the exam and what is not, your best friend is going to be a tool called the Curriculum Framework. And I have a link to that in the session description that you can see. So this is just a quick snip from the Curriculum Framework, just showing you how you can use this. So say I wanted to know, what do I need to know exactly about fossils? Just going to hit Control F on my computer. It's going to pop up this little find box, type in whatever it is you're interested in looking at, and then you can scroll through anywhere that pops up in the AP Biology curriculum. It'll describe what you do need to know. It's also, in some cases, going to describe what you don't need to know, which is really helpful. So if you have any specific questions, someone asks a question about which body systems you need to know, this is a great tool for looking that up. It also has at the very end of that document an index, so you can also look up specific topics here that you're interested in knowing about. Final tip here, if you're going to do some studying today, is about how to use your time when you're studying. Hopefully you all have honed some of your study skills by this point in high school, but when you're thinking about studying, it is way more effective as far as the use of your time to spend time testing yourself on things that you should know rather than just rereading notes or rewatching videos. So for example, if you wanted to study photosynthesis, I would first take a piece of paper, test yourself, write down as much as you possibly can remember, then use resources like a video or a textbook to check how you did, to fill in some gaps in your knowledge, and then test yourself again. All right, so just a few overall exam strategies before we dive more into multiple choice and free response. My first tip is that the formula sheet is your best friend. You're given it on the exam, so you should absolutely use it. The same with your calculator. Don't feel bad about using it even to test really simple math. Two things that the formula sheet is really helpful for. One is, of course, finding specific equations. There might be some equations on here that you're really familiar with because you've seen them a lot in class. I would still check yourself and use the equation as it's written on the formula sheet. One other way that the formula sheet's really helpful is that it often explicitly tells you what different variables in the equations mean. Again, even if you think you're pretty confident that you know what some of these different things mean, go ahead and look at the formula sheet, make sure you know what everything stands for. You can see an example here where they've defined a number of variables. It also happens down here. So just check yourself and make sure you're using the correct things. We're going to do one quick example. So this is a place where it'd be good to have some paper out and a pen or pencil. And this is just to show you that, looking at both pages of this formula sheet, there's a lot of stuff on here. There's probably some formulas that you've used all the time in class, some that you either never used at all, potentially, or just haven't used in a long time, don't remember very well. But I wanted to show you how a lot of the time, using the information you're given in a problem and the information the formula sheet gives you, you can solve a problem even if it's something you've never ever seen before. So if I got a problem like this for a grid in question, I would look over on the formula sheet, try to find something that matched up with the information I was given in the question. So in this case, it's asking me about diluting, and I see I have information about concentration, and I have information about volume. So if I look over here on the right, I see this formula with defined variables that's going to help me solve that. I might have never done it before, but I can probably figure it out. So I'm going to pause for like an awkward 10 to 15 seconds to let you try to set this up on your own based on the information in the problem. and then I'll show you the work that I did. All right, so it might or might not have been enough time for you, but to set this up, what I would have tried to do is look at my question and figure out how does the information I'm given match up with these variables that I've got here. So I saw that CI stood for my initial concentration, and it tells me I have initial concentration of 0.2. Doesn't seem like it gives me the initial volume, so for now I just leave that variable. It then tells me she dilutes the solution, ends up with a 2-liter solution of 0.1 molar. So I've got CF and CV, so my final concentration is 0.1 from right here. My final volume is 2 liters, take that from right here. So now I see the only variable I've got left is VI, the initial volume. So that's what I'm going to solve for. Sorry, I didn't realize I didn't actually put a question here, but that's what I'm going to solve for is the initial volume. So all I've got to do now is divide, multiply to isolate this variable, the initial volume, and I end up with 1 liter. So again, this is a type of question that maybe I've never seen it or used it ever in class or so long ago that I don't remember. But I shouldn't let that intimidate me because the formula sheet gives you a lot of information to help you. Last quick overall thing, although we'll talk about this a little more specifically with both multiple choice and free response, is that time is often a challenge for many students on this test. You only have 90 minutes for each part. We'll go over some strategies for how you can pace yourself in each section. One just overall piece of advice that I would give is to make sure you go to the bathroom before the exam. Sometimes you gotta do what you gotta do, but you really wanna have the full 90 minutes for each section because you're gonna need it in order to do the best you can on the test. All right, so we're gonna dive into looking at the structure of the multiple choice and some tips for the multiple choice. So, in terms of the structure, there are 69 questions on the multiple choice. You get 90 minutes, and it's going to be 50% of your exam score. 63 of those questions will be multiple choice, and some of those might be individual questions. That's what they mean by discrete. Some of them might be questions in sets, where you get a reading or a diagram or some data, and a set of questions will relate to that data. Then there are going to be 6 grid-in questions at the end, so we'll go ahead and look at some strategies for both of those sections. All right. In terms of pacing, 69 questions in 90 minutes ends up meaning about 1.3 minutes per question. That is definitely an average because there are going to be some questions that are really quick, really straightforward, and then some of those questions that come in sets are going to have a significant amount of reading you're going to have to do before you even get to the question. So I don't put that there to say that if you're behind that time for any given question, you're moving too slowly, but just in general so you know how to pace yourself. That means that when you hit the halfway time, you hit about 45 minutes, you would want to be done with approximately 35 questions in order to be on pace to finish on time. I know some people like to have more specific time markers than that. One other way to think about it is every 10 minutes, you should have done about 7 to 8 questions. Again, there are going to be questions that are slower and some that are faster, so you're not going to be right on pace with that the whole time. One other reminder is that you can't bubble after the 90 minutes is up. So whoever is proctoring your test should be giving you some reminders, especially at the end when you've got only 10-15 minutes left. With at least 10 minutes left, I would stop and bubble everything you've answered so far. Questions you haven't answered, you might even want to go ahead and guess. Pick C, pick whatever answer you want to pick, and then use whatever time you have left to go back to those questions you haven't answered, and do your best to answer as many of them as possible. But you definitely don't want to leave anything blank because you don't get penalized for wrong answers, so you might as well try every single question. And last thing is, don't get stuck on any one question for too long. As I said before, there are going to be some really, really challenging questions. Don't let those challenging questions hold you up from answering things that you're going to be able to answer more easily. So if a question's feeling really stuck, like you're not making progress, circle it, move on, and come back to it. Some other advice for the multiple choice. Don't be super intimidated by long readings and diagrams. You're going to see a lot of questions where they're giving you a lot of new information. That means it's information that you aren't expected to know yet, so don't be intimidated if you see things that you feel like you haven't learned. Trust that they're giving you the information along with what you know in order to be able to answer those questions. The best thing that I think you can do as you read those big paragraphs or look at those diagrams is annotate, figure out what you can tell based on the information they've given you, and then approach the questions. So we're going to look at one example of doing that. All right, sorry again if we lost that feed there. Hopefully it is back. Actually, you're going to find that you have all the information here that you need. So I'm going to be asking myself, what do these diagrams show me? So this first diagram I see is about mRNA. Second diagram down here is about proteins, one thing I see. I also see that this one is before fertilization has happened, and this one is after. And I see that bicoid seems to be associated, if I look at this diagram and this one, with the anterior end of the egg, and that this caudal mRNA initially is everywhere, dispersed throughout the cell, whereas after fertilization, it seems to be more associated with the posterior end of the cell. The last thing I notice when I look at the protein distribution down here is that their distributions don't really overlap. Only a tiny bit right here, but for the most part, bicoid is in the anterior end, caudal is in the posterior end. So if I've gone through and looked at these diagrams, identified some of these things ahead of time, it's going to make looking at the answer choices significantly easier, and it's going to let me cross a lot of them off. So last question I might have after looking at these diagrams is, why isn't this caudal mRNA, which I see up here dispersed throughout the cell, when I come down here to the protein distribution, how come it's not found throughout the cell? How come it's only found in the posterior? All right, so these were the answer choices you were given. I'm going to start looking at the bottom one and work our way up. So I'm trying to figure out which of these things best supports what I'm seeing over here in these diagrams. So D says caudal stimulates development of anterior structures. If caudal protein stimulates development of anterior structures, I would most likely expect to see caudal protein in the anterior. Based on what I've already noted up here and what I can see in the diagram, caudal protein seems to be much more associated with the posterior end of the egg than with the anterior end. So that's going to be an answer choice that I'm going to cross off. C says translation of bicoid mRNA produces caudal protein. If that were true, then I would expect that where I see bicoid mRNA, which has been translated into bicoid protein, I would expect to see caudal protein. And I noticed that I had caudal mRNA, but now I don't have caudal protein here. So that seems to be the opposite of what I see. Once again, I'm going to cross that one off. For answer choice B, if bicoid protein stabilizes caudal mRNA, again, that sounds like it's doing something positive associated with caudal mRNA. So where I have bicoid protein down here, I would expect perhaps that either caudal mRNA is still there, or at least caudal protein is there. And again, that's not what I see. So even just by process of elimination, none of those choices sound very good based on what I've already identified in my diagrams. But for the last choice, to make sure that one does fit, it says bicoid protein inhibits translation of caudal mRNA. So where I have bicoid down here, even though I had caudal mRNA, that mRNA is not getting translated into protein, so I'm not seeing that protein in my diagram down here on the anterior end. So I hope that shows you how you can look at questions that seem challenging, maybe are about something you haven't heard of before, and use the information they gave you to still answer them successfully. And definitely use the good old elimination method. There are going to be some choices that are more clearly wrong to you than others. And if it comes down to it and you're deciding between a few choices, again, you should still guess. You don't lose any points for guessing on questions. All right, for the grid-in section of the exam, this is the last 6 questions of the exam. Number 1 is that you should absolutely make sure you grid correctly. There will be directions reminding you of how to do that, but this is just showing you the multiple different ways that you can grid. It does not matter where you put your answer within these boxes, so you can start on one end, you can start on the other end. Totally fine, you can put your answer in the middle. Do make sure to grid in the negative if you end up with a negative answer. And you can put your answer in multiple forms as a decimal, fraction, but you should do so based on the information you're given. So every question is going to tell you at the end of the question how they want you to round. So, let me show you an example. So this question here at the end says, give your answer to the nearest tenth. That means that you should be showing one place after the decimal. Make sure you know your tenths, hundredths, and thousandths place. Because unfortunately, if you've calculated correctly, but you haven't rounded correctly, your answer will be counted as incorrect. So make sure as soon as you read the question, you circle what it's asking you to do and you give it in that form. Some questions are also going to direct you to give a specific type of answer. It might say percent, decimal, fraction, or something else. Make sure you give the form requested. In some cases, they give you a choice and say you can do it as a decimal or a fraction, in which case either answer would be counted correct. Last tip here is that I know most of you, at least for my students, are much more used to fancy graphing calculators, and you don't get to use fancy graphing calculators on this test. So what you need to do, what's really helpful to do with the simple 4-function calculators where you can't type in a giant formula, is to write down all of your steps. It's going to make you a lot less likely to make a mistake. It's going to help you see if you get an answer at the end that's not looking right. It's going to help you check your work. So I highly recommend, even if it feels like it takes a little time, writing down all of your steps as you go through your work. All right, one quick practice question we're going to look at just for the purpose of showing you. I think for my students, sometimes they think that the grid-ins are going to be the hardest questions and therefore maybe skip them or save them to the end and run out of time. Sometimes the grid-ins can be really challenging. Sometimes they can be very straightforward questions that you can answer pretty quickly. So if I look at this question, it's asking me to calculate the mean rate of population growth, and it even tells me the units. My units are individuals per day between days 3 and 5. And again, I want to make sure I get the correct information about how to round and give my final answer. So I know that a rate is just a change in something over a change in time. And they've even told me, again, it's a change in individuals over a change in time. So I'm going to find those 2 points on my graph, day 3 and day 5, as accurately as I can, and trace over to find the number of individuals, the population size that would be at each time. Then go ahead and do some quick math, find the change in individuals. So it's around 880 up here, 200 down here, and between days 3 and 5 would be 2 days. So I end up with 340 individuals per day. In this case, I got a whole number from my math anyways, but if I hadn't, I would have wanted to have made sure to round to 340 individuals. Just so you know, the vast majority of the time when you get a question like this with a graph, it can be hard to be really precise. That might not be exactly 100 or 880. They are going to give a range of correct answers. So don't stress out and worry that you have to have the exact number here. Get as close as you can, but they are going to give you a range of answers. All right. On to the last section of the exam, which is the free response. The free response, again, you have 90 minutes, same as for the multiple choice, and there are going to be 8 questions. 2 of them are going to be long questions. At least one of them will be lab or data based, and both of those questions are worth 10 points. Then questions, 3, 4, and 5 are going to be short free response questions, and those ones are going to be 4 points each. Then the last 3 questions are going to be also short free response. They're only going to be 3 points each. In terms of time, they typically recommend that on the long free response questions, you're spending about 20 minutes on each one. And for the short free response questions, you're spending about 6 minutes on each one. Again, the way you break it down might not be exactly like that, but you definitely should be spending more of your time on these long free response questions because they have more parts and they are worth more points. So there were a number of questions about this 10-minute reading period that your proctor is going to describe when they read the directions to you. So the first 10 minutes of the 90-minute free response section is called a reading period, but it is optional. It is recommended, but optional. So what that means is that they recommend that you use that time to read over all the questions, see what they're about, and plan out your responses before you actually start writing your responses. You can choose to do that or not. I think it is a good chance for you to prioritize. So as you look through the questions, you're probably going to find some that you are about topics you feel more comfortable with than others. And one thing that you can do is go through those 8 questions. At the top of each question, give it a number 1 through 8, based on the ones that you feel most comfortable with, which you would put as a 1, to least comfortable with, which you would put as an 8. And then answer the questions in that order so that if you do end up running really being rushed at the end, you're being rushed on questions that are questions where you maybe would have struggled more anyways, and you're not missing out on points that you definitely would have gotten on a question you were very comfortable with. The one caveat I would give to that is that you don't want to leave the long free response questions to the end, again, just because they're worth a lot more points. The other thing I'd recommend doing that time is brainstorming for some of the questions. Write yourself a little outline, write down keywords that you're going to want to include. It's going to help make sure your answers are more organized, and probably actually going to help you write more quickly once you start writing. A few quick tips. One is just to label each section. After each question, there will be prompts that are each labeled with A, B, C, D, E, F. Makes it a lot easier for your grader and probably easier for you as you're writing if they know exactly which part you're answering. And then you definitely want to prioritize writing something for each section. Say you have a long free response that has A through E prompts. You want to try to put at least something for each section rather than writing 3 paragraphs for part A but skipping the rest of them. Most sections are only going to be up to 1 to maybe 4 or 5 points at the most. So continuing to write more isn't necessarily earning you more points. But you know if you're skipping certain questions, like you don't write anything for C, D, E, and F, that you're automatically missing those points. All right. Let's see, actually, one other thing I forgot to put in here. There were some questions about how to list your responses. And you definitely want to write in paragraph form, but I want to clarify, since you all take English and history and other classes that might have very different writing styles, is writing in paragraph form just means you're writing in full, complete sentences. Does not mean you need to do a thesis and, X number of supporting sentences. You want to keep your answers as short and clear as possible, very specific to what the prompt asked. But that does mean you want to write your answers. Oh, I think I do have this on the next slide, but I'll say it now. Your answers not as bullets. You don't want to just put an outline. You don't want to just put a diagram. You want to use full sentences. Just like with a multiple choice, there's going to be some questions that seem really challenging. You want to use what you know plus what you're given in the question, and then just logic to figure out the best answer. You also really want to be specific with your language. If you're using the word it or they or food, the reader is not necessarily going to know what word you mean, and they're not going to be able to just give you the benefit of the doubt and assume you mean the correct word. So anytime you see yourself using something like that, ask yourself if there's a more specific vocabulary word you could replace it with. For example, if you're talking about plants and photosynthesis and you're saying they produce food, what you really mean is what they produce is glucose. All right. We're also going to look at an example free response question. All right, so as you look at a free response question, you really want to identify what you're being asked to do using the bold words in the prompt. They really help you by making these words stand out. And then as you've gone through and done each of those parts, I recommend physically checking it off as a way to really make sure that you've done everything that you've asked to do. Usually each of these prompts is going to make it really clear exactly how many points are in this question. So this is question number 4, which tells me it's a 4-point question. I see 4 bolded words, so I'm going to assume it's 1 point per question here. All right, so I've identified those words. And this one, we're just going to look at the first part of this prompt here as an example of a question where, once again, I hope you have learned about gene expression in your class. You might not have learned a lot about it, or come the exam tomorrow, you might not remember it easily. But you can still use what's given in this diagram here to get some points on this question. So part A says, primary transcript in the figure, which I see right here, is 15 kilobases long. The mature mRNA, which I see right here, is 7 kilobases in length. So the first question they ask is to describe the modification that most likely resulted in the 8 kilobase difference in length of the mature mRNA. So how come this one was 15 kilobases, this one was 7 kilobases? So I might happen to remember and have learned about splicing and be able to write about that right away. If not, I can just look up in this diagram and use what's labeled to help me write a good answer for that prompt. So I see this primary transcript looks very similar to this DNA up here. It's got these white sections, which are labeled as introns. It's got these black sections, which are labeled as exons. When I look at this mature mRNA down here, it doesn't have any of the white sections. It only has the black ones left. So I could write a simple sentence saying the introns were removed to make the mature mRNA. The introns were removed from the primary transcript to make the mature mRNA. I might not know anything about what an intron is. I might not know what that means, although again, I hope that you do. But just by using this diagram to help me and not assuming this is a question I can't answer, I could have earned some points on this question. All right, gonna pause here really quickly and just answer a few questions that have been coming up in your chat. One reminder, so if we're seeing a lot of questions about is X, Y, or Z blank topic going to be on the exam? One thing I'll say is that none of us knows exactly what's going to be on the exam. It's different every year. Your teacher, no other teacher out there has seen it, so we don't really know. But in terms of what things could be on the exam, the best thing for you to do is look at that document that I shared earlier, called the Curriculum Framework. It's inside the course and exam description. And the link to that document is in the notes right under the live stream. Again, if you just hit Control F, you'll be able to search that document. Say you're interested now in reviewing about introns, hit Control F, type in introns, hit Enter, and you'll find all the information that could be tested on the exam about introns right there. All right. We're getting close, guys. Stick with me. One thing I would love to see more of, haven't gotten too many ideas from you all yet. Again, you all have taken a lot more tests more recently and frequently than I have. If you have strategies, please share them out, and we'll share them out here with everyone. So if you look through free responses that have been released, and there are a number on the website, I don't think that link is currently in the notes under the stream, but I can add that for you if anyone wants to go look at some example free responses. You'll see that there's some really common prompt words that are those bold words that indicate what you should do. So you might get words like predict, propose. All right, sorry, I think you might have lost the feed again, but we should hopefully be back. And again, we are looking at common prompt words for the free response. Two very specific types of things you might be asked to do. One is to calculate. If you're asked to calculate, one quick piece of advice I'll give is make sure you include units in your final answer. And then we'll look really quickly at an example graph for things you should include if you're asked to graph. Although as a reminder, there was a whole session yesterday on lab skills in the exam. And there's a link to that schedule, so you can go watch that review if you missed it yesterday. The final types of words that are pretty common are these words like justify, explain, or provide reasoning. If you get questions like that or a prompt like that, this is where you probably are going to want to write more than one sentence. They're asking you to say, based on either data you've been given or information in a diagram or what you already know about biology, to explain your answer further and say why something is happening. If you write a sentence for this and you're able to still ask yourself, well, why? Why is that true? How do I know that? It's an indication that you should probably write another sentence giving a little bit more of an explanation. For graphing, so you very well could be asked to graph something, especially in one of those big questions, question 1 or 2. These are the main types of things you want to think about as you're doing a graph. You want to have axis labels that are very specific, and we'll look at where you can get those from. You definitely want to include units for those labels. You want to make sure your scale on the x and the y-axis is even. Use a key if you need it. So for example, if you have two different lines that you're graphing, make sure you use a key that differentiates which line is which. There's usually a point given just for making sure you properly plot your points or your bars if you're doing a bar graph. And then some questions are going to ask you to draw error bars. If so, they'll give you information about the standard error of the mean or 95% confidence interval. If they give you that, you should graph it. So here's an example, I believe, from the released 2013 exam, which is available to you online. Gives you this data table up here. There was also some accompanying information, some reading that I left out. And it says on the axes provided, create an appropriately labeled graph to illustrate the sample means of the three populations to within 95% confidence, and they tell you that means the sample mean plus or minus 2 times the standard error of the mean, which is listed right here. For the rubric, in terms of how they actually gave points for this question, they gave 1 point for having a correctly labeled, scaled graph with proper units. So you had to have both of these things in order to get that first point. In terms of a correct label, I want to identify my independent and dependent variables to know what goes on the x and what goes on the y. And then I would recommend, if you've got a data table like this, just using exactly the label it gave you and the units it gave you. And I'll show you an example of what I mean of that in just a second. Then you could have drawn either a bar graph, or you could have actually just plotted points. We'll take a look at that. And those had to be done accurately. And then you had to show your error bars 2 times above and below the mean. So here's one student example that earned full points. So for correct labels, the student just took trichomes per centimeter squared from this right here. I probably would have written the full thing out and written trichome density, and then put this as the units in parentheses. And then on the bottom, you're looking at plant population, so they just wrote plant population and listed populations 1, 2, and 3. Along here, they have an even scale, so they get that full first point. They have appropriately plotted their means for each population. And then their error bars go 2 above and 2 below, so that's 2 times the standard error of the mean, which is just 1, for all 3 populations. So that would be an example of a graph that would earn full points. All right, let's see what other questions we have gotten here. So, a student asked, is there a break between sections? I believe there definitely is a break. Maybe a moderator out there could correct me. I believe it's about 10 to 15 minutes that you get in between the sections. Please, someone out there correct me if that is wrong, but you definitely do get a break between the 2 sections. And you'll only have 1/2 of the test at a time, so they'll collect the multiple choice from you before you get the free response. Someone else asked about labeling each section of the free response. You should definitely label. They're not going to take off points if you don't, but keep in mind that these graders are grading a lot of tests, and anything you can do as far as labeling, having neater handwriting, just to make it easier for them to read your answer and identify where the points are, is going to help you. And then, oh, yep, I see that it is a 10-minute break. So in between the 2 parts, you get a 10-minute break. If you want to see a full released exam, just to look at what the directions look like and what the exam looks like in case you haven't done a full one in class, if you just Google this, and I'll include the link because I'm not sure if it's there right now, the 2013 Practice AP Biology exam, that is a full released exam that was given in 2013. And you can see the entire thing, you can see what the directions look like. Looking through that just might help you feel a little bit more comfortable come Monday. All right. And I see one student idea out there that we have that is a great suggestion, which is if you're going to do any studying today, again, not late at night, make sure you are sleeping, but if you're doing any studying this afternoon, one great resource to use are the Bozeman Science videos. Shout out to Paul Anderson for making all of those. There's actually a link in the notes under the stream to that if you want to check it out. All right. So, those are actually all of the tips and strategies I have for you from the exam. The only thing I wanted to add quickly as far as miscellaneous review, because it was a question that came up from a number of people, was about photosynthesis, which is always a hard topic. I will again remind you that there's a link under the live stream to all of the review sessions, one of which focuses on Big Idea 2, which would include photosynthesis. It would include probably more detail than I'm going to go over now, so go ahead and check that out if this is a topic you're interested in reviewing a little bit more. All right. So first, probably the most important thing at this point that you can study is the big picture of photosynthesis. So what is its purpose? I'm thinking about the equation. Where do things go in and out? So, excuse me, hopefully we know the equation for photosynthesis. Let's see, I can write it back here for us. Hopefully you can see this whiteboard. So for photosynthesis, plant's going to be taking in carbon dioxide and water, going to be producing glucose and oxygen. So looking at this diagram, we'll give you a few seconds to try to identify for yourself, for those two reactants and two products, where are they going into this process and where are they coming out of this process? All right, so for my two reactants, which would be the two arrows coming in here, water is going to be the reactant coming into the light reactions, which includes the electron transport chain. The reason water is being used here, oh, sorry if it's hard for you to see on the whiteboard back there. Water is being used as a source of electrons. So when the water comes in, it's actually going to be split by the light, so that we have a source of electrons for the electron transport chain. When the water is split, it's going to produce both hydrogen ions and oxygen, and that oxygen is just going to be released as a byproduct of this process. For our other two reactants and products that we have, carbon dioxide is going to be coming into the Calvin cycle here. It's going to help us produce the carbohydrates that are eventually going to help us produce glucose. So the other main product over here is eventually going to be glucose from the Calvin cycle. You can also see internal to this process things that are being recycled between the light reactions and the Calvin cycle. So they're not written in this overall equation because they aren't overall reactants or products. The light reaction is going to produce ATP, which is going to provide energy to the Calvin cycle because that can happen all the time, not just when there's light. It's also going to produce NADPH, which is an electron carrier. It's going to bring electrons from the electron transport chain over to the Calvin cycle. When both of those things get used, when they've been used in the Calvin cycle, we produce ADP plus a phosphate and an ADP plus. Those are going to be recycled back to the light reaction so they are ready to be used again. All right. Very last thing about photosynthesis is diving in a little more detail to the electron transport chain, which is in the light reactions. One thing I want to say in looking at a diagram like this, or probably the diagrams you have in your textbook, is that you do not need to know all the names of all of these little proteins along the way. And that's something, again, that if you look in that curriculum framework document and you looked up photosynthesis, it would explicitly tell you what you do need to know and what you don't need to know about photosynthesis. The things I would focus on in reviewing this is trying to figure out how are these two products of the light reactions that I know are necessary to drive the Calvin cycle, how are they produced through the electron transport chain? So, first step we're going to look at is how we produce this electron carrier up here, NADPH. So as we said before, as a reactant of photosynthesis, we need water. So water is going to be split by light as a source of electrons. That's where we produce that byproduct of oxygen. Those electrons are going to move down the electron transport chain. And when we say move down the electron transport chain, we mean they're moving from one protein to the next, to the next, to the next, starting at photosystem 2, ending up at photosystem 1. And what causes them to move is electronegativity. So if one molecule is more electronegative than the next, it has more of an affinity for electrons. So for example, this one right here is going to be more electronegative than photosystem 2. So the electron is going to be pulled from photosystem 2 to this protein pulled to the next one because it's more electronegative, pulled to the next one. Eventually, they end up at photosystem 1. They're given another jolt of energy by light that's still being received by the chloroplast. And then that electron is going to be added on to NADP plus to end up making NADPH. Again, the whole job of NADPH is an electron carrier. So it's picking up these electrons at the end of the electron transport chain and bringing them over to the Calvin cycle to be dropped off. So we got this part. Now we got to figure out how we make ATP. In order to make ATP, as these electrons are moving from one molecule to the next, again, they're moving from one that is less electronegative to one that is more electronegative. As they do that, they actually they lose a little bit of energy. That energy is going to be used to pump hydrogen ions or protons, which are always just floating around in the cell. Those are going to be pumped across the membrane. You can see one crossing right here using active transport. That's going to be building up a concentration gradient of hydrogen ions or protons in the lumen of the thylakoid. So it's going to be more acidic over here than it is on this side. Those hydrogen ions, can't just pass through the membrane on their own. They need a protein channel to pass through. So this molecule at the end here is called ATP synthase. That's going to provide a channel for the hydrogen ions to go flowing through. They're going to come rushing through. As they go down their concentration gradient, that energy is going to be used to phosphorylate an ADP and end up making ATP. So again, if I were testing myself on this, I'd start with that big picture of what's going into each part of the photosynthesis, what's coming out of each part, and then I would try to figure out in the light reaction specifically how is ATP being produced, how is NADPH being produced. There's a lot less detail that you really need to know about the Calvin cycle, but again, check out that curriculum framework if you want to know more. All right, so we're going to end with a last word of wisdom that we got from Ms. Dixon's AP Biology classes in Rockland, California. And they said, remember, at a cellular level, you are quite organized, and as a living thing, you're part of the biology that you're going to be tested on. So you guys have got this. Breathe deep and smile big. And I think that is great advice to end on. Again, you guys have learned so much this year. You're here on a Sunday studying, like you're doing an awesome job. So look over some of these pieces of